So now let's switch over and talk about how the data looks and the data object that you'll be receiving to work with. So two packages that will be relevant for you by way of introduction are ScanPy and data. ScanPy is a very commonly used package in Python in the single cell community. And data is a package within that that is specifically very useful for dealing with these single cell gene expression matrices and the associated metadata that they might have, such as the perturbation identity that we've been discussing. And so here I've just provided some very kind of initial code that you might want to use to import some of these packages and work with their functions. So for example, import ScanPy as SC, and also NumPy and pandas, which are useful for data frame manipulation and statistics. And so going a little bit more into the data package. So you will be able to use it to read in the data that we'll provide to you. So with this function sc.read h5ad. And if we actually then look at what the resulting data object is here, you can see that it's made up of multiple objects. But the two really key ones in here are, first of all, this green matrix X, which you can access by writing adata.x. And so this is the single cell expression matrix that you will be familiar with by now and that was generated from the Perturb-seq experiments. And the other particularly relevant object here is this obs object, which you can access by adata.obs. And this will give you associated metadata for the cells in the expression matrix. So we can see every row here is a cell, and we can see in the second column the perturbation assignment. So again, whether this was, let's say, MLX-IPL that was knocked out, or another gene, or a non-targeting control. And there's another column that we'll provide, which will be the assignment of cells based on high expression of certain programs. So for example, you can see a differentiated adipocyte program that was high in cells 2 and 3 and 5. And so diving into these objects a little bit more, we can see, first of all, this just tells us the shape of the adata.x object, so the gene expression matrix, which should correspond to the number of observations, i.e. the cells times the number of variables or features, which are the genes. We can also use this to show just the first few cells and genes, so the first 5 cells and genes expression values in the adata.x matrix. Similarly, we can use adata.obs_names to see the names of the first 5 cells, or the unique cell IDs. And finally, similarly, we can write adata.var_names to see the names or the IDs of the genes that are, again, the first 5 columns in the matrix.